The SAGE, the SAGE come up as one of the most promising organization of Central India. The SAGE is led by a visionary chairman, Engineer Sanjay Agarwal ji, who has been awarded more than 50 awards at national level and has a reputation of owning best engineering college in group by MP. To know more about our group, I request Dr. Neeraj Jain sir to play the corporate video, and I request everybody to pay attention to our corporate video. Thank you everybody for your kind attention. Today's Sage Winter School is has the theme of computer-aided drug design, and yes, sir. So, am I audible?
Yes, ma'am. You are audible.
So, and computer-aided drug designing has its own importance and has its own value in the coming future. For this, we have with us Dr. Gaurav Kumar. Sir, I welcome you on behalf of our group, and thank you for sparing your valuable time. And it's my pleasure to introduce you to all the participants today, Dr. Gaurav Kumar. Is currently working as assistant professor, program chair, and research and PhD coordinator in the clinical research department, School of Biosciences and Biomedical Engineering, Galgottias University, Greater Noida, UP. He did his PhD from School of Biomedical Engineering, Indian Institute of Technology, Varanasi, UP. Previously, he served as assistant professor at Shobit University, Gangaon, Saranpur, and assistant professor at Shobit University, Meerut. Lecturer at Sardar Vallabhbhai Patel University of Agriculture and Technology, Merit and research trainee at CSIR, that is Scientific Instrumentation Organization, Chandigarh. He was selected for the DST Inspired Fellowship in 2012. He received gold medal in Masters of Technology, that is Biotechnology, in 2012. Outstanding Reviewer Award from Elsewhere Publication in 2018, and Best Faculty of Biotechnology Award from William Research Center, Tamil Nadu in 2020. He has published 35 research review papers in reputed journals. He has published 5 book chapters, Springer and Bantham Science. His major area of research includes neuroprotection, drug discovery, and wound healing. He is member of editorial boards of Frontier Neurology, EC Neurology, and Journal of Biological Engineering Research and Review. He's also an active member of several reputed scientific societies, including SFN, IBRO, IAN, SNCI, and IEEE. He has organized several national and international conferences, seminars, workshops in Shobit University, Gangaon, Shobit University, Meerut, and Galgottias University, and has presented his research work at several international and national conferences in India and abroad. Thank you, sir. Thank you for joining us and
Thank you.
sparing your valuable time, and thank you for guiding us students as well, sir.
Thank you.
Over to
Thank
you,
you
sir.
so much, ma'am, for this elaborated introduction.
My
Thank you
pleasure.
so much for providing me the opportunity as a speaker in this event. So, what I know, I would like to make learn the participant, and this is the beauty of teaching. So, dear participants, student, you are free to ask any query during my presentation also. I would like to start from scratch, means from the beginning. I think several participants does not know about the drug. So anyone can tell me about the drug? What is a drug?
तुम रहोगे ना?
Lodhi, yes, you are not audible.
जा रहे क्या?
Yes, yes, yes.
All
Please.
participants are requested, please turn on your video. So there is an interactive session with the Gaurav sir.
Thank
We
you.
have only 31 participants in this session for this Sage Winter School. So there is a limitation. So all please turn on your video.
Have joined. Yes. Okay. Anyone? Can you explain about the drug?
Yes, sir.
Okay, okay. What is a drug?
A drug is a chemical entity which is used to prevent, cure, or treat diseases.
Okay. Yes, you are right, but 50% right, because some anesthetic compound is also regarded as a drug. And they do not have any therapeutic value. So we can define a drug as a medicine or any compound or other substance that can play a physiological effect when it is administered or ingested or introduced into our body. So drug is any substance that have a physiological effect on our body. So in this lecture, I think I can share my screen. Okay, yes, thank you. So it is visible? Is it visible?
Yes, sir.
Okay, thank you. So I am going to briefly discuss about the structure-based drug discovery using molecular docking, because I am not very much expertise, but I have published about 7 papers on the basis of molecular docking. And the people are working in the Schrodinger software, which is a highly valuable and most costly software in the world. And they are not able to publish a single paper with in silico study. But you can publish. So if you want to learn, if you want to do the study, you can able to publish in SCI journal. Yes. So if you will learn, then you can publish. So now I am going to start first of all about the understanding of the drug discovery process. So when we start from a scratch, that if you want to identify or if you want to design a drug, then it is not called as a drug. It is called as a ligand or a small molecule. And there are several steps that can executed by the researcher to translate that small compound or ligand into the drug. So first we have to understand the drug discovery process. So it has four steps. First is research and developmental stage that encompass about 3 to 6 years. It consists the first thing like target identification. I will let you know about the target. So target identification, and to know about your target, it is very important step. If you do not know about the target, then you cannot proceed for the drug discovery or drug designing process. So in the next slide, I will elaborate on the target. Now compound screening. So you have to make a library of small molecules. That molecule can be obtained from the various source, animal source, phytochemicals. It can be a phytochemicals. It can be hormones, endogenous molecule. It can be a peptide. It can be monoclonal antibody. So there are several sources of the small molecule that can be translated into the drug. Now, after screening of the compound by using in silico analysis, actually in silico analysis is the study that that is performed with by the help of computer, and it is it attracting up the attention of the scientist for the easy screening and cost reducing strategy for the drug discovery process. So after that, so you have to install some software for the compound screening, and after that you have to identify a lead molecule from that library. The second step of the drug discovery process called as preclinical study, and the preclinical study, you can say the in silico studies there, in vivo studies there, and in vitro study. So in vitro study is performed. I am sure that you just know about the in vitro study. So it is performed in the test tube as well as Petri plates. You can culture a cell. You can also perform the enzymatic reaction. That is also called a in vitro study. In vivo study is performed in the rodent and non-rodent model, and it is also performed in the human subject also. But in the preclinical study, the human subject cannot be involved. So in the clinical study, you can involve the human, and it comes under the in vivo study. So then you have performed the in vivo study, and to test the efficacy as well as toxicity, you have determined the ADME property of your molecule. And if your molecule is efficacious, then you can go for the clinical study. But before translating your molecule into the clinical study, you have to require some standard. So minimum you have to perform 2 types of study, one in rodent model and another in non-rodent model. Non-rodent model, for example, the rabbit, cat, dog, and higher level of mammals, but not human. So after that, when you the regulatory body pass or screen your protocol, then you have to perform the clinical trials. The clinical trial also has 4 phases. In this diagram, phase 0 is not mentioned. So first is phase 0, phase 1 trial, phase 2 trial, and phase 3 trial is there. As well as after phase 3 trial, your drug can be marketed, but it is limited to some practitioner, medical practitioner. After that, you have to done the regulatory body have done some review and approval process. You have to submit several type of document for that can evaluate the post marketing of the drug. And if your drug is causing any adverse event during the phase 3 trial, it can be withdrawn from the market also. So there are several example of the drug that has been withdrawn from the market on the basis of their adverse reaction. So adverse reaction is any unwanted reaction that happened after administration of the drug. After that, if your drug is very efficacious in the human subject, as well as it cannot, it has less side effect, but most of the drug which available in the market has some side effect. You cannot neglect the toxicity level. So there are several factors that is associated, but it cannot be a part of this lecture. So after that, you have to post-release monitoring process. Pharmacovigilance is also performed. So here is the phase and process of clinical trial. Here you have performed the preclinical study using in silico, in vitro, or in vivo study. You have to validate the drug efficacy in the in vitro sample or study, or in the animal. You have to assess the toxicity of your drug. After that, you have to file the investigational new drug application, INDA. And after that, when it is approved, then you can translate into the clinical phases that encompass phase 0, that is microdosing. Here only about less than 15 subject or human subject can be recruited, can be enrolled for the study. And here the main goal of PI or principal investigator is to test the drug toxicity level on the human patient. So the drug, whatever you have translated into the clinical trial, then you have to inject a small amount of the drug to the human subject. If here adverse reaction is occur, then your trial cannot be proceed further. If your drug is passed in 0 phases, then your drug will be translated to phase 1. That evaluate the safety. Here in the phase 1 also, the safety concern is monitored. If your drug is determined as a safe, then it can be translated into the phase 2. In phase 1, there is also 20 to 80 people is involved, that is recruited by the various clinical center. After that, phase 2, in the phase 2, the more number of participants should be recruited on the same day order, on the basis of the mode of action of your drug. So here there are 2 type of concern. The principal investigator is testing their effectiveness as well as the safety. So till phase 3, the safety concern is measured. So while using in silico analysis, our lecture, this lecture is based on in silico study. So for understanding the importance of in silico study, I have just giving you some brief idea about the translation of the drug. So Now, when your drug is effective, showing effectiveness and it has evaluated as a safety, then it will be translated into phase 3. And in the phase 3, the effectiveness is also is again confirmed. The monitoring of side effect, means adverse reaction, is also performed and compared to the other treatment. Here, your drug can be compared to the other treatment. For example, if any drug is existing for the treatment of same disease, then it can be compared with that standardized treatment. So not a drug is only tested in the clinical trial. You can also perform clinical study for the process, for the surgical process, for the implantation of any microchip or any assistive devices. For example, pacemaker. If you have created any pacemaker, if you have created any 3D type of organ, then you have to perform the clinical trial. And in the phase 3, usually 3,000 participants should be participated. After that, it go to the FDA review, and you have to file new drug application. So new drug, when NDA has been approved, then your drug will be translated into the phase 4 trial. After that, if all things is right, then your drug will be in market. So it is taking about 5 to 15 year. Some exception is there also. For example, COVID vaccine. So here is the emergency condition. So regulatory body have some flexibility to perform the clinical study as a fast track, and you can get your drug in 1 year or 2 year also. Now we are moving for the identification of a drug. So if you have any query, then you can stop me and you can ask your question. Okay, now, so first, our aim is to identify a drug that can modulate, that can inhibit the action of any target, that can activate any protein action. So for example, you have to, if you want to identify or design any drug, then you have to first define your problem, which disease you want to treat. So first you have to read the pathophysiology of the disease, and you have to identify the target. So if you want to identify a molecule that can protect, for example, neuron in the disease condition, okay, so then how it will be happen? So you have to search the literature, the second step, because literature, there are numerous literature is available on NCBI PubMed database, where you can search, and you can also Google, and you just read some SCI indexed paper, not go for the any predatory journal paper. So so just read some paper that is published by the reputed publisher like Elsevier, Springer, Nature, Cell Press, Science, Thomson, yes, Thomson, sorry, Taylor and Francis, IOP, and all the things. So now, so if you know about the exact pathophysiology of the disease, for example, you want to treat any brain-related disorder, you have to treat any diabetic type of disorder, then you have to study the pathophysiology of the disease. Then you can identify a target. A target can be a protein, it can be a DNA, it can be a RNA, it can be a ribosome that can be modulated by the drug. So for example, you have chosen a protein because most of the drug is targeting protein. So so protein has several forms like receptor, channel, symporter, antiporter, enzymes. So first, when you have decided that our target is protein, then you have to learn about their three-dimensional structure, their mode of inhibition, their mode of activation, whether it should be activated, whether it should be inhibited. So you have to learn about the role of target in the modulation of the disease pathology. For example, your protein is overexpressed in the disease condition, and that lead to devastating condition in the disease. Means the disease is caused by the overexpression of any protein. Then your strategy will be to inhibit the action of that protein. And if any disease is occurred due to the down regulation of any protein, then your strategy will be the activation of that protein. Simple. So how should I proceed? How you will proceed? So first you have to know the structure of your target. Then you have to identify the pocket, active site, their catalytic site, or their allosteric site of the protein. Now you have to recognize the critical residue that is responsible for the modulation of protein activity. And here PDB, Protein Data Bank, will give you a great insight to learn about the three-dimensional 3D coordinate of the protein, as well as their inhibition as well as activation strategy. So if you identify the site where you want to see the interaction of your molecule, then you are ready to proceed. So please keep in mind that when you are going to opt in silico study, you have to do this is a theoretical type of work. So you have to do more and more extensive literature survey to justify your outcome, your result. So you have to discuss your result more extensively. Now, next. So what is the molecular docking? Actually, molecular docking is a method by which you can analyze the conformation and orientation, that also referred as a pose of the molecule into the binding site of macromolecular target. So for example, protein binding docking is a study of how a small molecule, that is called as a small molecule ligand, bind perfectly with the macromolecule or protein. That can also be a target. The searching algorithm generated possible poses which are ranked by the scoring function. And there are several software were developed by the bioinformaticians during the last decade, among which are some well-known examples such as Autodoc Vina, Doctor, Gold, Flex, Molegro, Virtual Docker. So you can opt out, you can choose any one of them. You can also, if your institute has funding, then you can also purchase the Schrodinger, because Schrodinger cannot be purchased by an individual until analysis they have a lot of money. Because when you want to subscribe a Schrodinger software, then it can be a cost of approx 15 lakh per annum. So for 1 year you have to charge 15 lakh rupees. Now then why should we wait for the Schrodinger like of software? So a true researcher is that can do research without the resources, and there is no limitations. So you can choose accordingly. There are several software which is freely available, and and when you produce the result by that software, sorry for the disturbance, 1 second. Hello up
Gaurav sir, are you there?
Yes, sir. Yes, sir. I'm just coming.
Okay, okay.
Oh, okay. Sorry for the disturbance. Actually, one person I want to know about something. Okay, so we cannot wait for the costly software, and we can perform the molecular docking or structure-based drug discovery using freely available software. So for the molecular docking, you can identify the potential inhibitor or activator by using this. So you can predict also the mode of action of a compound. So there is reverse engineering also. For example, several drug is prescribed by the doctor as a off-label. Means a drug is approved by the some regulatory body like FDA or SIRTSP for treating a disease. That if that drug can be prescribed to treat some other disorder, then it is called off-label drug. So doctor knows that this drug is effective. For example, rheumatoid arthritis, a drug is developed or approved for to treat the rheumatoid arthritis, but it can also be prescribed by the doctor to the patient of, for example, diabetes, and doctor knows that the drug can be efficacious in the diabetic condition, for example, then you can know the exact mechanism of the drug by molecular docking, how this molecule, this drug is modulating the target of diabetic pathophysiology, means the molecular mediator of diabetic pathophysiology. Now, so there are several type of drug target. For example, ion channel, transporter. It can be a transporter, it can be an enzyme, it can be a receptor. So the drug target can be a DNA, drug target can be an RNA, it can be a ribosome. So tetracycline, you heard about that. The tetracycline is a effective antibiotic that is blocking the amino acyl transfer RNA site, A site, by binding on the A site. So there are several type of drug target, and you can choose that target according to your procedure. So now, why we are going for the drug target identification? So drug target identification is the process of identifying the direct molecular target, that molecular mediator that involved for the disease progression, and to identify a biological target that should be a druggable, and a term is called druggable if its activity can be modulated by a therapeutic, whether it will be a small molecule, a drug, or a biologics. So the technique that used may be based on the principle of biochemistry, biophysics, genetics, chemical biology, or other disciplines. So practically every medicine is interacting with a molecular target, either boosting or inhibiting its function. So most of the drug, when you study about the any drug, available market drug, then when you want to study, then you will realize that most of drug belongs to the inhibitor category. like acid channel inhibitor, it can be histamine inhibitor, it can be DPP4 inhibitor, it can for the treatment of diabetes, for the treatment of ulcer. There are several type of drug that has inhibition effect. Some drug also work as a activator, so you can also activate the drug. Sorry for the disturbance. Once again, it is I'm again back 2 minutes. Please provide me 2 minutes.
Okay, sir.
Okay. So till now, any question? If you have any query, then you should ask. No? Then should I proceed? Now, what is the property of a
Sir, excuse me,
promising
sir.
drug target? Yes.
One query is there. Shall I ask?
Yes. Yes. Yes.
How to identify a target for a particular drug, sir?
Yes. First of all, you have to decide your problem, means a disease. You have to select a disease. After that, you have to study about their pathophysiology using literature. Or it is the very basic thing that you can know about the drug by by reading the literature. Also, you can perform several type of assay to identify a target molecule. If the target is not yet established, it means, for example, hydroxychloroquine is widely prescribed during the COVID-19, and a researcher and doctor claim that it is helping to treat for the treatment of COVID-19, for example. But no one know what are the actual mode of mechanism of hydroxychloroquine for the COVID-19. So when you want to know about your target, then you have to perform several type of assay. You have to study the pathway of the drug. You have to crystallize some molecule, some protein that is that can interact with that drug. So I'm just I will refer you, and I will recommend you to, if you are new researcher, then you have to perform extensive literature survey for the clue of the target for the drug discovery in initial stage. And then when you will be sound in the research, then you can identify a receptor, you can identify enzyme that can be a possible drug target or therapeutic target. I hope I have addressed your query.
Yes, sir. Target means a particular disease, right?
No. Disease, no. in a disease, I will let you know. I will address your query in later, and you will get sufficient information in the next slide also. So disease is not a target. In a disease, when you will study the pathophysiology of the disease, how the etiology, causes of the disease, so then you will know about there are several type of molecule, means molecular mediators like protein, like DNA, some receptor, some enzyme that play a devastating role. Their overexpression, their down regulation is related to the disease progression. That the target should be a protein, it can be a DNA, it can be a RNA, it can be an ribosome. So I will cover your query in the next slide also. So hope you should you may understand about the you can differentiate about the disease and target. So target can be a protein molecule, it can be a DNA molecule, it can be RNA molecule, it can be a ribosome molecule. Okay.
Yes, sir. Yes, sir.
So the
I got
target, when I address about the target, so target should be a protein, it can be a DNA, it can be RNA. So you have to clear-cut idea about that. So when I told target, yes, someone told target means a therapeutic molecular mediator that can be modulated by a small drug, or a small molecule, or any drug. So the target has a confirmed role in the pathophysiology of a disease, the disease progression that should be directly linked with the pathophysiology of the disease. For example, brain ischemia happen when NMDA receptor is overexpressed. The blood-brain barrier is disruption or brain edema happen when the exaggerate activity of MMP2 and MMP9. So I will cover in the next slide also. Okay. Now there are some property of promising target that should be kept in mind. That target has a confirmed role in the pathophysiology of disease. The target expression is not evenly distributed throughout the body. For example, if you identify that any protein that has direct link with the disease progression, but you have to ensure that that protein should not be expressed in all cell of the body. So target should be specific. If you will inhibit any protein that is playing a vital role, for example, CY450 enzyme that is expressed in the liver, when you are targeting nitric oxide synthase enzyme, so be careful during that. Nitric oxide synthase has several variants, so you have to specify that there are 3 types of variant of nitric oxide synthase: endothelial nitric oxide synthase, inducible nitric oxide synthase, and neuronal nitric oxide synthase. So your drug molecule, if you are targeting any neurological disorder, then your drug cannot inhibit the INOS, or it cannot inhibit the endothelial nitric oxide, because the endothelial nitric oxide synthase is expressing in the endothelial lining of your capillary, and your molecule when interact with that one, then patient can show adverse event and their blood pressure can be enhanced. So you are going to target any neurological disorder, but your drug is doing some adverse event. So target cannot be evenly distributed throughout the body. Now, you have to keep in mind that the target 3D structure should be available to access their druggability. It is very convenient process when a three-dimensional structure of protein is available in protein data bank. Irrespective that you also perform molecular docking when 3D structure is not available in protein data bank. You can do homology modeling, or you can predict the structure by the threading. So, but the three-dimensional structure which is available in the protein data bank, and one structure which is predicted by homology modeling or threading, cannot be as similar, and it can give a better result. So it will be better that your molecule, your protein should be available in protein data bank. If not available, then you can perform the homology modeling. There is no issue. The target is easily accessible. Accessible means you can perform, you can assay their mechanism of action by in vitro study, by using in vivo study. For example, if you want to inhibit any enzyme, you should buy that enzyme in pure form, and you can perform your reaction in a test tube. So the target is easily accessible. That is also a characteristic of therapeutic promising target that enable the high throughput screening. And the target poses a promising toxicity profile, and potential adverse effect can be predicted using phenotypic data. So there are several databases available. There are pathway database is available that by using that database, you can know more about your target. So I am again repeating, when you are going and you want to publish a paper using in silico study, only computer-based study, then you have to do more literature survey. You have to gain a wide knowledge about your target that can be defended during the publication of the paper. So the proposed target has a favorable intellectual property status. You should know about that. You cannot patent an idea. Idea generally cannot be patented. But when you have identified any drug that can act on a enzyme, kinase, or any receptor, and that show efficacy in vitro model or in silico model, then you can file a patent. So that drug should be patented for the 20 years. And only you can transfer that drug, you can sell your patent, or you can manufacture that drug after that discovery. So there are two types of strategy for the target identification. I am addressing your query. There are two strategies. That is for the target identification. You can go for the target deconvolution, as well as you can go with the target discovery process. So there are two types of strategies. Target deconvolution process. In this scenario, you begin with a drug that appears efficacious, and the target is identified retrospectively. Means you know that an off-label drug, that for example, I am again repeating that one drug is approved by FDA or CDSCO for the treatment of diabetes, but some medical practitioner or doctor is recommending that drug also for the treatment of rheumatoid arthritis. But the physician cannot know and not able to justify how this drug is beneficial for the treatment of rheumatoid arthritis. Then you have to find your target, where your drug is going to bind, and it is interacting with which type of molecular mediator. That process is called target deconvolution process. And target deconvolution yes, for performing the there is a lot of noise.
थोड़ा सा मुझसे ले लेता नहीं दे रहा होगा मैं क्या बक रही नहीं दे रहा है
Host, please. Host. Okay, sir. Nilesh sir, please mute.
वो मेरी डिग्गी आ गई
Bit to C.
क्या
Hello, Nilesh sir. Okay.
एक्चुअली क्या है, वो नेटवर्क का इशू था तो मैं कंट्रोल नहीं कर पाया, वो बिट्टू सिंह का बटन दब गया होगा।
Okay. Okay. So
बिट्टू प्लीज म्यूट योरसेल्फ और कोशिश करें कि आप ना अनम्यूट नहीं हो।
okay. No, he can unmute himself when you have any query. Okay. So when you want to deconvolute any target, or if you want to identify a target by the target deconvolution process, you have to work in any established lab that should have some sophisticated type of instrument like X-ray crystallography, X-ray diffraction study, and NMR, electron microscope, and all the things. So this process can be done in any reputed lab. But you can go with the target discovery process. The target discovery is very easy process. In that approach, you have to identify your target first. And if you want a new drug, you must find a new target. So, for example, if you want that, if your hypothesis is that ashwagandha can be that there are several type of phytochemical present in ashwagandha, or any herbal plant that can act as that can be administered, or that can be used to treat any disorder, then you have to study that disorder pathophysiology, and you have to identify the target by using literature. So once the target is identified, the compound library should be made, and it should be screened using molecular docking mechanism. And after that, you can perform the molecular dynamic simulation study. Now, the drug discovery strategy. For example, there are 4 types of scenario. Like, if you know the protein structure, means protein structure here is placed and ligand structure is placed here. Now, if you know the protein structure that is already deposited in the protein data bank, and you know the structure of ligand that is already deposited in PubChem database, drug bank database, and zinc database, then you should go with the structure-based drug design. That can be done using the docking and scoring or virtual screening. If the protein structure is unknown, but ligand structure is known, you know the ligand structure, small molecular structure, but protein structure is not deposited in protein data bank, and you cannot able to find in the protein data bank or any other protein database, then you have to perform the ligand based design, drug design, that consists pharmacophore designing or QSR study. When the protein structure is unknown, as well as a small molecular structure or ligand structure is also unknown, then you have to perform the STS, high throughput screening or combi chemistry, matlab combinatorial chemistry or virtual screening. By using combinatorial chemistry, you can generate 1,000 to lakh of the small molecule, or you can synthesize the peptide. You can, by using high throughput screening, you can perform the assay of multiple assay in one time. If the protein structure is known and ligand structure is known is unknown, then you will perform the de novo designing like virtual screening. So this is the flow chart by which you can choose the in silico study. Now, I have told you about the small molecule and ligand. So, for example, you...
Excuse me, sir.
Yes, yes.
One question is there. Shall I ask, sir?
Yes, yes, of course.
If we have newer molecule, newer structures or
Yes.
newer derivatives, then which docking will prefer? Structure based or ligand based?
Means if you have new molecule.
Yes, yes. We have synthesized the new molecules. Suppose.
Okay, you have synthesized new molecule and you know the structure of that molecule or not?
Yes, yes.
You know, no? Then protein and protein structure is also available in protein data bank. Then you should go with the molecular docking and scoring function or virtual scoring function.
Achha, then
Yes.
it comes under structure based or ligand
Yes,
based?
yes, it will come under the structure based drug design.
Okay, sir.
But if you do not know the structure of the ligand, if you do not know the structure of the protein, then you should perform the so protein, if protein structure is unknown and ligand structure is known, then you will perform the ligand based drug design, that is pharmacophore screening process, QSR study. Any other question? Should I proceed?
Yes, sir.
Okay. Now I have discussed about the small molecule. So I think that all participants know about the target. Any difficulty will be sorted out in the next slide. So a small molecule, you know, you can prepare the small molecule using synthetic chemistry. You can also generate the small molecule using combinatorial chemistry. You can also generate by monocular hybridoma technology. So that is the your ligand molecule, but we cannot called as a hit molecule. When we should called a small molecule to the hit molecule. So there is some differences. When, for example, your target molecule is, for example, nitric oxide synthase. You want to inhibit the action of nitric oxide synthase. Then, but you have listed possible small molecule. For example, you have 100 small molecule. And that small molecule cannot, the 100 small molecule cannot be regarded as a hit molecule. When you will perform any in vitro study, when you will perform any in silico study, or when you will perform any in vivo study, then you will come to know that the 100 small molecule cannot may not be so efficacious in for the same assay, but there are some 2 or 3 small molecule that so efficacious or that can interact with your target molecule. Then that 2 or 3 molecule can be regarded as a hit molecule. So hit molecule identification can be carried out by using virtual screening. You can perform the docking study, you can perform the de novo designing, scaffold hopping, fragment-based screening, building blocks, STS, phenotypic screening, all will give you an idea for the hit identification. When you have identified a hit molecule from your small molecular library, then hit optimization process can be needed or cannot be needed. Hit optimization process is done to change any structure of your molecule. For example, you can see a small molecular structure of any compound, a 3D structure of any compound or 2D structure of any compound. Then that compound may have some functional group. So all the structure of a small molecule cannot be utilized and cannot be interact to your target directly. The portion of a small molecule that is interacting with your target is called and responsible for the modulation and responsible for their effectiveness is called as a pharmacophore molecule. And that pharmacophore, apart from that pharmacophore, for example, this is the small molecule and this portion is responsible for the action, but this portion causing some adverse event, some portion cause some toxic effect, some portion are responsible for the low absorption value when administered via the oral route, then you have to perform the lead optimization process. So you can change, you can alter the structure of your lead without hindering their pharmacophore. That is called hit optimization process. So the many times optimization of hit is not needed, but when your molecule is showing efficacious, but it also showing some toxic effect, it also showing some low ADME profile, then you can change the structure of the molecule. That is called lead optimization. But after hit identification, you can identify your lead molecule. So when you have determined your lead molecule, then there is a need, then you can perform the lead optimization process, and otherwise you will go for the clinical study. And after performing the clinical study, phase 0, phase 1, 2, 3, your small molecule, your lead can be regarded as a drug. So there are 3 words: ligand, small molecule, one category, the hit molecule, the lead molecule, and the drug. So you cannot call drug to the lead molecule. You cannot call drug to the hit molecule. Now, I think you have understood. Any query? So there are some basic steps in silico drug design. For example, it is also called computer-aided drug discovery. So CAD is it is also referred as a CAD. So for example, if you are working on a disease, that disease-related genomics study have been previously done, or you can also perform some bioinformatics study for identification of any gene, any homology modeling or something. So the CAD give you some scope for the target identification, for the target validation, for the lead discovery, for the lead optimization, and you can perform the preclinical study, means in vitro or in vivo study, and you can go for the clinical trial. Now, for example, I am going to give you an example. For example, neuroprotection, related to the neuroprotection. Neurological disorder is the most promising disorder, and it is the leading disorder. Is cerebral stroke is the third leading disorder for the morbidity and mortality in the human. So if, for example, ischemic, for example, if you want to develop any drug in the ischemic stroke, so there are, I have listed some target that should be targeted in the ischemic stroke. For example, Cluent 2B containing NMDR, caspase 3, caspase 9, MMP2, MMP9, mu and M calpain, PRP1, ROC in ROC 2, INOS, NNOS, RIP1 kinase, and for example, if you are going to discover any drug for the Alzheimer's disease, there are some target: NMDR, beta secretase, cholinesterase, tau protein kinase, BAS1, gamma secretase, GSK3 beta, histamine 3 receptor. So, and if you are going to identify any drug for the treatment of Parkinson disease, there are some target is also listed here. For example, NMDA, you can see that NMDA expression is overexpressed, or it due to their abnormal activity, it can cause a wide disease. Means it is involved in Parkinson, it is also involved in Alzheimer's, it is also involved in ischemic stroke. Yes. Okay, again bit 2. Okay, so Parkinson disease. So in the Parkinson disease, you can inhibit the action of alpha-synuclein, CABL, glucocerebrosidase, glucosyl ceramide, LRRK2, but you can also activate GLP-1 receptor, MCL1 receptor. So I have listed this target molecule on the basis of the literature survey. So you can also, if you want to identify any drug that can be developed as a skin ointment or for the wound healing, for the diabetes condition, there are a lot of diseases you can pick as per your interest. So here, when you will, for example, here I am giving you some example. For example, ischemic stroke, because I have done my PhD on the ischemic stroke. So when you will, when I want to discover, I want to identify a molecule that is present in ashwagandha or Withania somnifera or green coffee. Basically, so in the green coffee there are several molecules there, and what molecule, which molecule is actually working as a molecular mediator, means which molecule is actually working to combat the disease progression. So you have to identify first the problem identification, means I will work on the ischemic stroke. Then you have, when you will search the pathophysiology, when you will extensively search the literature, then you can be able to understand the pathophysiology, and I have listed some of the molecular mediator which is represented in the red box. So for example, I want to make you learn about the pathophysiology of ischemic stroke. In the ischemic stroke condition, ischemia is nothing but a deprivation of oxygen and nutrient to the brain tissue. So when you heard the word of ischemia, means the deprivation of oxygen, there is hypoxic condition is there. So when the hypoxic condition is there, the glutamate concentration will be raised, and the extensive rising of glutamate concentration can activate the NMDA receptor. When NMDA receptor will be activated, the intracellular calcium level will be increased, and the calcium is the most potential a small molecule that can modulate if their activities cannot be controlled and their homeostasis is not established, then it will create a devastating, it can play a devastating role in the pathophysiology disease progression of ischemic stroke. Not only in ischemic stroke, you can see NMDR overactivation can also play role in Alzheimer's disease, Klinefelter-Jakob syndrome. It can also be cause in Down syndrome, Parkinson disease, Huntington disease. In all diseases, NMDR activation should be regulated. So first target, your NMDR can be your first target. After that, even you will proceed the related consequence of NMDR activation means how the calcium is playing a devastating role for the neuronal cell death. So here the calcium can activate the myocalpin, it can also activate the iNOS and nNOS, it can also causes the mitochondrial dysfunction, and the mitochondrial dysfunction that can lead to the activation of caspase-9. It can also generate the oxidative stress, it can also cause the nitrosative stress, and due to that stress, apoptosis inducing factor will be raised and the cell will go for the apoptosis by the PARP activation, and ultimately what will be there? The neuronal cell death. So when you will study the pathophysiology of any disease, you can find enormous number of the target molecule. So the participant who has asked about and they have confused about the disease and target molecule. So so I hope that they have understand what is the target and what is the disease. Any query, or should I proceed? Any query? No? Okay. Am I audible?
Yes, sir, you are audible.
Okay. Now, so I am leaving this one. So for example, if you want to identify an inhibitor that has higher affinity toward the target compared to the reported inhibitor. So while performing any study, you have to pick a reference molecule or control molecule also. For example, if you want to inhibit the DPP4 that is overexpressed during the condition of diabetes. So DPP4 inhibitor is already available in the market. So for example, beta-gliptin. Beta-gliptin is usually prescribed by the doctor to the diabetic patient for the treatment of for the lowering of blood sugar level. So actually, when you want to identify a novel drug or a new drug that can inhibit the action of DPP4, then you have to compare your docking result with the control molecule, for example, beta-gliptin. So when your docking score will be better, your molecule has a better affinity toward the DPP as compared to the reported inhibitor, then you can report your study. Because you have to justify your study, why you are reporting, why you are identifying any novel drug. However, the drug is already available in the market. So there will be two reasons. First reason is you want to identify, you want to perform your research, and another reason can be the adverse reaction of pre-existing drug. So you have to also list the adverse event or side effect of pre-existing drug that will be advantage for your study, for your justification. So now we will I am proceeding further. For example, you have to perform several method, and you have you required several tool and software. For example, molecular docking software, you can download the Autodock. I will make some hands-on training if time will be permitted. Then we have enough time for the hands-on session also. I will briefly because I have about 40 minutes. Nilesh sir?
Yes, sir.
Okay, so I will make you learn some software, how you can install the software, what are the software that is needed for the drug discovery process. So I'm recommending Autodoc software. It is very easy to use and freely available software. You can find it. It is freely available. You can easily download this software, and it has not a requirement of a very high configuration of computer. But if you do not have at least 4 GB RAM in your computer, in your laptop, do not do the docking, because I have uh, uh, my 3 laptop is um, not working due to the molecular docking. So this is our 4th laptop in my research area, because so you have to pay something. So you have to buy or you have to increase your RAM first of all. If your computer has 4 GB RAM in our latest version, then you can perform the molecular docking. If you had 1 TB of hard drive, there is or SSD, then you can perform the molecular docking software, because during the energy minimization process, there is a lot of memory is used, and that can crash your hard drive. So it will be better when minimum requirement is you can perform with the i3 processor and 4 GB RAM. At least 4 GB RAM should be there. Now, visualization software, you should use Chimera. Chimera is very convenient to use, and there are several type of function available in the Chimera software, and it is also freely available. 2D ligand protein interaction viewer, link plot, you can also download, and it is also free. The small molecular drawing software, Marvin Sketch, is available for the academic purpose. You can use this software also to draw any structure of the molecule. You can design a new drug without synthesizing in the laboratory. So by in silico, you can use this Marvin Sketch or ChemExon software or ChemDraw software. You can also use. You can also use several NCBI also has some online server by which you can generate some molecular structure. So for the energy minimization of the small molecule, you should use the Pyrex. Pyrex is also available freely, and Chimera, I have told earlier. And for the, if you are using Windows, then you have to download the Syswin. So Syswin will give you Unix-like environment for the window. So Syswin installation is a critical step, and by using Syswin, you can perform the molecular docking. Now, so you need some database web server for the analysis. For example, Protein Data Bank, PubChem Zinc Drug Bank. These 2 databases are not freely available, so you have to pay money for the retrieval of the small molecule. But PubChem is freely available server, and the phytochemical. If you are sorry. Sorry. Okay, phytochemical screening, Dr. Duke phytochemical, Dr. Duke have developed a database for the phytochemical screening. We will learn about all the things. The drug likeness prediction, you can perform the drug likeness. For example, pre-admit moles of are freely available. The blood-brain, if you are dealing with neuroprotection, then you will go with the blood-brain predictor, or there are several server is also available. Now we will learn about Marvin sketch. We will learn about UCSF Chimera, Pyrex, Autodoc, Sysbin, LeafPlot. So these are some software is needed when for the better illustration, for the generation of better illustration and better understanding of molecular docking. Now the retrieval of three-dimensional structure, first step. So you have to retrieve, or you have to generate the three-dimensional structure of a small molecule. So here I am going to stop the sharing and sharing the screen, whole screen. Okay. If you have any query, then you can ask me. After that, I will proceed further. So,
Sir, excuse me, sir.
yes, yes.
Shall I ask?
Yes, yes.
Sir, these softwares can means we can run in Windows 7
Yes, yes, you can run. Of course, you can run.
or Windows 10
No,
compulsory required.
no. You can use in XP version also, but RAM size should be large. You can use in XP. Huh? Then, yeah, Windows 7 you can use. Now, first of all, PubChem database. How many participants know about that database, PubChem? How many? Okay. So there are some participants. You can raise your hand. Okay, only Raju Varghese. Okay. Okay. So you just search the PubChem in Google. PubChem. You just search, it will appear. So PubChem is a database of the chemical database. It is freely available, and you can see here that my screen is showing or not.
No, sir.
Okay, sorry. Okay. And now
Yes, sir.
okay. PubChem. Okay, you just search on Google. Go with the PubChem. Now PubChem is the database developed by the National Library of Medicine, NLM, or NCBI. So NCBI, you heard about the NCBI. Okay, I think, because you have bioinformatics and something, so you must know about the NCBI. So it is a database provided by the NCBI. So it is also regarded as called as PubChem NCBI database. So here you can see that there are more than 110 million compound is deposited, and about 277 million substances there. The bioactivity of this molecule is available about 293 million. The literature you can also find the link, how you can find literature in the PubMed Central and PubMed database. But here the small molecule, their bioactivity, which is linked with the literature, so about 34 million literature is direct link of literature is available. 28 million patents is also available. So you can just search any molecule, for example, paracetamol, so you just search and you will get the structure of paracetamol. It is also called acetaminophen. So here in the paracetamol structure, you can get the PubChem ID. This is important ID. When anyone is identify or design a new substance by synthetic chemistry, and he know about their structure, they can deposit their structure in the PubChem database, So there are some review status is there. When you will submit your structure, that structure and your literature will be sent to the some reviewer, and you will get some reviewer comment. After that, when it will be accepted, your structure will be published in the PubChem. For example, you can also submit your gene structure. You can also submit in the gene bank, NCBI nucleotide data bank. You can also submit the some research paper that is also indexed in the PubMed Central. And here the PubChem CID is unique CID, which is given to each and as a unique identifier for each and every chemical. So here there are 2 or 3 type of structure will be available. Two-dimensional structure, there is 3-dimensional structure. 2D structure is generally used to draw, to represent the illustration of your small compound, but for the molecular docking study you have to download the 3D conformer of the molecule, your molecule. So you can download here from SDF. You can also download the structure, get image, a small, medium, and large size, better resolution image. So for SDF, the structure data file, you can download it, and you can also open this molecule in Marvin Sketch. Now I am introducing PDB data bank. So protein data bank is also freely available. You can search any of the molecule, for example, any target cache page. For example, cache page I want to know, then cache page 3, for example, apoptosis related cysteine protease. So in the PDB, you have to keep mind some things related to the when you are performing the search. And I forgot to told you some feature about of PubChem also. So here you can find the title and summary. So in the PubChem database, when you visit or surf the PubChem site, then you will know about the synonyms of the molecule, molecular weight, and their about the molecule. You can also get the literature, some 3D dimensional structure, crystal structure is also there. Now you can select the name and identifier, and you can get the UPEC name, INSKI, and canonical SMILE, which is widely used for the prediction of their ADME toxicity profile. So you can get the molecular formula also. You can get the chemical and physical property, what is the molecular weight, and all the several things is related with one molecule. So you can see here the medical information, which company will supply this molecule also listed here. Some agrochemical information is there. The pharmacology and biochemistry is also associated with that molecule. So you can get several type of information using PubChem database. Now PDB. So in the PDB data bank, PDB data bank is also a public data bank that consists about it consists about you can count here about million, no 18 lakh structure of the protein is deposited here. 18 no 1 lakh 86,000, sorry, 1,86,000 protein structure is deposited in the protein data bank. And if you are searching any protein, then you have to keep in mind that what will be your model. Means the protein is deposited from various sources. various organisms like Bos taurus, like Mus musculus, like Ratus novargicus, like Homo sapiens. So there are several sources. If you need some human protein, then you have to identify here for their structure. If you want to perform your study in rat, then you have to identify the molecule of caspase or any protein that belong to the that can be extracted or expressed that can be isolated from the that organism, means from the rat or from the mus musculus. So now this is the protein data bank, and in I'm just clicking protein entry, then you will find out several type of identifier. First is the PDB ID. This is the one unique ID which is assigned to each and every entry. 1,86,000 protein is deposited, and each protein has a unique identifier. So here three type of a study that are eligible if you are performing the X-ray diffraction method, if you are performing to predict the three-dimensional structure of the protein by using NMR solution structure, or electron microscope. If you are using electron microscope to predict the three-dimensional structure of protein, then you there are three type of tools, technique that can be utilized for the prediction of, for the determination, not prediction, for the determination of the three-dimensional structure of protein, and it will be deposited in PDB. And there are several processes for the deposition. When you have identified, for example, you cannot identify here human structure of this is the zebrafish caspase 3. So caspase 3 is not available. Caspase 3 is not available for the human. So for example, if you want to isolate a caspase 3 from the human, and you have performed the X-ray crystallography study to determine the three-dimensional structure, you can deposit here. After deposition, after submission of your structure, there will be review, and after implementing or review or comment, your structure can be published here. Now our goal to show the structure here, you can easily find out by which method your structure has been determined. So here X-ray crystallography structure, the structure determined by the X-ray diffraction method, it is called as a crystal structure. The when you go with the NMR structure, this is called the solution structure. So their resolution, their R value, their organism, the expression system which is utilized, if there is mutation or not, when it is deposited, whom this structure is deposited, all the information, and the literature associated with that protein, which is very useful for the determination, for the understanding of structural organization of that protein. That I will recommend, strictly recommend you to read each and every line, and you have to understand this paper very carefully when you are going to design any drug, and when you want to dock your drug into that protein domain. So you have to determine the structure, or the catalytic residue or allosteric residue, the amino acid, the pocket in the active, which is present in active pocket or catalytic pocket or allosteric pocket. So that molecule should be interact with that amino acid. So you have to clearly you have to read this literature, which is freely, it can be freely available on PubMed Central. So you can just download and read this structure. If your target is caspase 3, then you have to read this literature very carefully. Now, the second thing is caspase 3 has two chain A and chain B. So you have to where you will perform your molecular docking study, in chain A or in chain B. So that information should be that information will be provided by the author who deposited the structure in the PDP. मैम हेलो मैम, हाँ मैम एक क्लास ले रहा हूँ, मैं आता हूँ कुछ देर में। हाँ बोलिए क्या एरिया का लॉगिन नहीं, उसका पासवर्ड मुझे दिया था क्या? मैं ढूंढ के बताता हूँ आपको, ठीक है, लेकिन मुझे लगता नहीं दिया था मुझे, मैं देखता हूँ। ओके ठीक 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 ओके ओके हाँ सॉरी। Now you can know the sequence length, how many amino acid is there in that chain, in which organism this protein is derived, there is mutation or not. So you will prefer the mutation. You will not prefer the structure that consists any mutation. So their gene name is also associated. Now the main thing is that here is there are D and C chain is also there, and there is no mutation is present, and how many ligand is associated with that structure. So you can know all about by reading this this literature. So now next yes. Now I am going to elaborate some server. You can note it, for example, Swiss ADME toxicity database. So Swiss ADME profile, if you want to learn any molecular profile, physical property of any molecule, then you have to go to the search the structure in PubChem database, and you have to go with the name and identifier. For example, you have to pick the canonical smile. You just copy and paste here smiley. Now you will run the program. Now it will generate some data. After that, it will generate some data like their physiochemical property, their lipophilicity value, their sorry, lipophilicity value, their water solubility, the pharmacokinetic value, means how the GI absorption is high or not, it is lipophilic or not, it is blood-brain barrier permeant or not, it is CYP inhibitor or not. So be careful when you are going to design any molecule. When you are preparing a small molecular library, you have list, for example, 10 molecule for the docking, then you have to perform this, you have to predict their pharmacokinetic as well, and clearly check that is your molecule can be predicted as an inhibitor of CY450 enzyme, then you cannot proceed with that molecule, because if any molecule can inhibit the action of CYP450 variants, then it can cause a hepatotoxicity. So you will not continue with that molecule. Now, the most important feature is Lipinski rule of 5. Lipinski LOR, it is also called as LOR. So Lipinski have established some rules for the drug discovery. And there are some rules like a molecule should have a molecular weight less than 500. A molecule, the molecular log p value means their lipophilicity value. This one, mole log p value is also it should be below 5. There is hydrogen acceptor and number of hydrogen bond donor, that should be less than 10 and 5 respectively. So, and their molar refractivity value should also less than 50. So Lipinski rule of 5 is not having 5 rule, but all the clauses, all the condition of Lipinski rule of 5, the value of Lipinski rule of 5 can be divided by the 5. That's why it is called Lipinski rule of 5. So if your molecule is predicted pass the Lipinski rule of 5 means yes, with zero violation of the rule, then your molecule can be translated for the clinical setup, and it can be it has potential to be developed as an oral drug. So Lipinski rule filter give you a most extensive information about your drug. So if your molecule is violating the Lipinski rule of 5, that means that do not mean that your molecule cannot be translated to the clinical setup, but it can cause toxicity. It can cause it can not permeable to the blood-brain barrier. It can not be absorbed by when you administer the drug via the oral route. So there are several obstruction is there. So prefer those small molecule that can have zero violation with Lipinski rule of 5. Now, any query? No. Now, I am going to, sorry, yes. So 2 hours is not enough, but sorry. So one also software is there. No, this one. So if you want to, so just note or take a screenshot for the PKCSM. It is it also has several type of features about a small molecule. So you have to put the smiley here. You just copy and paste the smiley of molecule here and just paste here and go with the ADME profile. So here you will get several type of information. So here you will get the EMS toxicity. For example, if your molecule are predicted as mutagen or not. So it can cause carcinogen, or it is predicted as carcinogen or not. So you can get the information from the this server. So is your molecule is creating hepatotoxicity or skin sensitization or not. So there are several type of factor that can be predicted on the basis of the smiley file or on the basis of their structure file. So there are several server available that can help you to determine the drug likeness property, blood-brain barrier permeation property, hepatotoxicity profile, ADME profile, and for your small molecule. Now, I am not presented yet the molecular docking. Now that we have retrieved the 3D structure, so now I briefly I am telling you about the Marvin sketch. So you have to download the Marvin sketch, and by using Marvin sketch you can generate a small molecular library. It is very easy to use. So you have, for example, you can randomly generate some molecule. You can give you can generate the molecule. For example, this one, you can change the residue. For example, I do not want CH, then you just put here Cl. So and this molecule can be cleaned in 3D mode. So just clean in 3D mode and save a file as a PDB. So it will generate a 3-dimensional structure. So you can save, you can generate a lot of molecule using Marvin sketch. You can also draw any structure, and you can clean any structure, convert any 2D structure to 3D structure by using Marvin sketch, or OpenBabel is also there. Now, next, any confusion? So I think you are exhausting.
No, sir. Please, can you demonstrate how we convert this 2D structure into 3D structure?
Yes, you just
Marvel
download
project, basically from B Form first year to 8th semester and M Form. So they can hand it notes to the how to use the Marvel structure sketch.
okay, and so you have used already.
Mhm.
So it will be better now. You just download the SD structure data file from the PubChem and just open to the in the Marvin sketch. You just open in the Marvin sketch. For example, I have downloaded 2D structure. For example, just you open that molecule in this one. And after that, you just go with the structure clean 3D. So you just clean 3D when you perform clean 3D and just save the structure, save as in 3-dimensional file. It will generate a 3D structure, very clear structure. No. Okay. Now, after retrieval of 3D structure of a small molecule from the several databases and retrieval of protein data, the 3D structure of protein, you have to optimize the structure of protein as well as you have to optimize the structure energy minimization. You can perform the energy minimization by using Pyrex also. You should perform the drug property prediction and molecular docking simulation. In the molecular docking simulation, you have to install required software, for example, Autodock, Chimera, Pyrex, Sysbin, LickPlot. After that, the file conversion of ligand and macromolecule, grid formation is there, docking preparation is there, so and docking is there. So docking and protein ligand binding analysis you will perform according to that. So I will make you learn very briefly about Autodock. So you have to first you have to make the library of small molecule. For example, synthetic, and the nature of a small molecule can be synthetic, phytochemical, endogenous peptide, and you can list the molecule like in a table what you want to show to the reviewer. So now you have to make the small molecule library by using Dr. Duke. Okay, I have forget to mention the Dr. Duke phytochemical database. So there are several phytochemical database. Because why I have included the phytochemical? Because you will wonder to know that in 20 year earlier 20 year, the FDA approved drug that encompass about 21% or 23% drug directly comes from the phytochemicals. So FDA approved drug is has originated from the phytochemical. So there are several people are want to explore the Ayurvedic formulation, want to explore several type of medicinal plant phytochemical to understand the mechanism of action of that the active constituent. So you have to perform you have to screen some molecule. For example, if you want to work on Bithania somnifera, Bacopa moneri, or Curcumina longa, that is curcumin is there. Guruchi. So you just go to the Duke Duke phytochemical database and just type, for example, Bithania somnifera. So here you will get the result, and you can exclude the ubiquitous chemical from here. So because ubiquitous chemical is founded is the chemical that can distributed in each and every plant, so we should exclude the ubiquitous chemical. Here you can get the result. For example, there are 150 molecule. You can screen the molecule with their higher ppm. For example, betaferrin A, some bethanolide D, bethasomnin, beta-sitosterol, and all the things you can list, and you can prepare your library from this here, and you just search their structure in the PubChem. For example, I am going to search the structure of first molecule, bethapherin A. So bethapherin, I have just searched bethapherin A. Now bethapherin A structure is available. So here I will go to the 3D structure of bethapherin A and just download the structure of SDF format for the molecular, ready to molecular docking. Now, the next step, the 3D structure retrieval of a small molecule, I have already covered. Okay, so determine, this is the critical step to determine your reference molecule, because for the comparison. So molecular, if you want to perform molecular docking and identify any molecule from the docking study, you have to compare your binding energy result with your reported molecule. So for example, you have to prepare some chart that can have molecular weight, rotatable bond, total polar surface area, hydrogen bond donor, hydrogen bond acceptor, log p value, log d value, log s value, and all that etc. From the Lipins, there are server developed by the IIT Delhi supercomputing facility of bioinformatics for the calculation of Lipinski rule of 5. You can use the MolSoft, you can use Swiss ADME, you can use the PKCSM, you can use the BMDRC, you can use online blood-brain barrier predictor, and from the PDB you can download the three-dimensional structure of the protein. Now, what we will do if the protein structure is not deposited in PDB, then you have to do the homology modeling. So as time is not permit me to elaborate the steps of homology model, so I am just skipping this one. So these are some publication that solely published in SCI journal by molecular docking for your realization. So this is me as a first author. This is published in Springer, Interdisciplinary Science, Computational Life Science, and this is published in Bentham Science, later in Drug Discovery and Design, and this is published in Journal of Theoretical and Computational Chemistry. It is also a CI journal. This is also published in Journal of Theoretical and Computational Chemistry. We have also published in Springer Nature. Thank you. But I am just briefing you the molecular docking step. If sir will permit me for 15 minutes.
I request you, sir, please spare us 10 minutes more to how the docking process is going on with the software. Please explain.
Okay. So should I give 2 minutes break to the participant?
No need, sir. Please continue.
That is needed for me actually.
Okay.
Yes, sir.
No problem, sir.
Okay. So dear participant, I'm come back within 2 minutes. So please do not move else. Okay. Now I am coming for within 2 minutes. Okay. Thank you.
All participants, please, I request you to stay connected with us. Sir have demonstrated how docking process is going on with using a software.
Okay, sir.
Meanwhile, I will explain you something related to the today's Sage Winter School. Sage Winter School, the main objective of that will provide the computer-aided drug design, the basic principles and the techniques involved in that, and the software which are used in CAD. In the whole program, 5-day program, there is some assignments and quiz. The quiz will be floated by in a Google form. So you have to just, there is a multiple choice question, you have to answer them. And depending upon the evaluation seat, we have to provide you the certificates. The attendance is compulsory for all 5 days. So I request all to pay attention for that. The MCQs are basically the content which are provided by the speaker. So please note down everything, whatever they explained by the speaker. The third day, fourth day, and fifth day. Basically, there is a 3-day, we have a quiz and then an assignment. The total marks of there is evaluation sheet is a 50 marks. So anything you have to want from my side, please ask me. Okay. Tomorrow we have a another speaker. There is an eminent speaker from the Sodinger. He is a trainer, basically from the Vinod Devaraji. So he will explain the how we use the Sodinger software. They provide the free. They will provide the free link for the Sodinger software using a small. Matlab, there is a limited accessible content and descriptive models in the Sodinger. So you have to take a screenshot or take the picture of that link. I will provide the whole video of that particular lecture in your mail ID. So you can download from there also.
Sorry for the inconvenience. I am back.
No,
So
sir.
are you ready to learn?
Yes, sir, please.
Okay. So we have demonstrated about the Marvin sketch. So now you have to download the Autodoc software. So for example, you have downloaded the 3-dimensional structure, PDB structure from the PDB, and you have downloaded a ligand. So I am going to demonstrate you aldose reductase is also a therapeutic target in the diabetes. So for example, if you want to identify any anti-diabetic drug, anti-diabetic lead molecule, then you will perform your target may be DPP4 or your target may be aldose reductase. So for example, you have downloaded the structure of aldose reductase. Now I am going to download in front of you. So is my screen is visible or not? Okay, no, not now.
Sir, participants want to know that Autodoc software is freely accessible with all.
Yes, freely accessible. It is freely accessible. No cost is I am just giving a demonstration how to download the Autodoc. So first, so you have to install the MGL tools for the window, for example. Okay, so you just type MGL tool that is developed by the Script Research Institute. So when you go with to the download MGL tool, you have to download the MGL tool first from the download page. Okay, from here. Screen is visible, sir?
Yes, sir. Yes, sir.
Okay. So here you have to download first MGL tool. It is the latest version of tool. If you are your operating system is Windows, then you just click here. If you are operating with Apple, then you can use this one. So for the Linux version, you can download this one. So now the lower version you can also download. So first you have to download the MGL tool. After that, you have to install. After that, you just download Autodoc 4.2, for example, this one. So when you go with the Autodoc, MGL is a Python based programming that will run your Autodoc. So you have to download this one, Autodoc for the window. If you are operating with the window, then according to your operating system, you will download the file and you will install the software. Now you will search for the UCSF Chimera. It is also available freely. So just search UCSF Chimera and download the Chimera also. Now this is the Chimera. So for the Microsoft window, you just click here, .exe file will be downloaded and you have to install the Chimera also. Now we have downloaded 3 software. Now you should download the Marvin Sketch by searching and ligplot. Ligplot. So ligplot homepage, here you have to do the registration process and the license key will be delivered in 72 hours. By the when your application will be approved, they will send you a link to download the ligplot. So for downloading the ligplot, you have to do registration, and you can able to generate this type of files, means interaction file. For example, this bond is related with the hydrogen bond, this is the hydrogen bond, and this residue is in hydrophobic interaction. So you can justify your result in the discussion section, that how many residue, critical residue for has interaction with our molecule, with the hydrophobic interaction, and how many hydrogen bond is formed during in the catalytic site or active site. Now this is the ligplot download, and you can download the Sysvin also. Sysvin download. Okay, so it is also needed for the window operating system. If you are your operating system is Linux based, then there is no need to download any Sysvin software. You can directly give the command from the Autodoc. So now, first of all, I just giving a small demonstration. For example, aldose reductase. So from the scratch, okay. So you will go to for the molecular docking with the Autodoc software, you have to perform in C drive. First point. So first you will go to the C drive. There will be user, there will be your admin. Then in the admin file, you have to create a single string file name. For example, I have created a folder with name of Autodoc. So you can also create a folder with name of docking. So here, be careful. You should not give 2 names. For example, Autodoc my, for example, this. So your software will not run. So here you have to give only single letter file, or you can use you should not use the space or something dash like that. So you keep a single string file name in the C drive. So you will go C drive, user, your admin, and then you have to create an Autodoc. So in this folder, you have to paste 4 type of file that will be retrieved when you will install the MGL tool. You will go to the C drive program file. Then here the Script Research Institute, you will find the Autodoc, and in the Autodoc you have to copy these 2 files and you have to paste these 2 files in your user, admin, and your Autodoc file. So is it clear or not?
Yes, sir.
Okay. So and these 2 files will be available in the that folder also. So you have to keep these 4 files in your folder. Now, second important thing, that if you have 100 molecule, a small molecule, then you have to create 100 folder with the name of that small molecule. Be careful, because when you will XYZ, you will or you when you are maintaining some Excel sheet, then you can put the file name like one, that can encompass to that can correspond to the paracetamol, second file number 2 that can chlorophenicol, file number 3. So or you can create a folder with the name of the small molecule. This is needed for the better resolution, for the better, for minimizing the confusion. For example, I have docked some compound like azithromycin, clarithromycin, LCDAR, or erythromycin, vinoverlin. So I have this small molecule. For each small molecule, you have to create one folder in the Autodoc or your whatever you have, your folder name in the C drive. Now it is clear.
Yes, sir.
Okay. Now, okay. So you just go to the PubChem, for example, PubChem, and here my small molecule, name any a small molecule, for example, erythromycin, no, any
Indol.
paracetamol I have also downloaded. So so I will dock with the paracetamol. Okay, PD, for example, you will go with the PDB. So you just search your molecule. And my hypothesis is that can paracetamol inhibit the action of aldose reductase or not? For example, aldose reductase. So you just put the aldose reductase name in the search bar, and here you will got several type of molecule. Aldose reductase from the barley, from the butanol, from this. So there are several types of structure is there. For example, if my target name is neuronal nitric oxide synthase, then you have to type NOS, neuronal nitric oxide synthase. You have to type NOS. So there will be result for the neuronal nitric oxide synthase. So mutated heme domain, you should not proceed with the mutated one. So for example, here several type of species structure is available. For example, Ratus norvegicus, Homo sapiens, Bos taurus, Drosophila, Mus musculus. So just if you want to do docking with the Homo sapiens protein, then you have to select the Homo sapiens protein in the filter section. So just filter it and proceed. So all the structure will display here with the relation that is isolated from the human. So for example, I have taken this structure. You just download the PDB file, and from the bottom of the page, you will get several type of domain ligand here, that is heme domain. And why I have told you that you have to study the structure of the protein. So I know about the structure of NOS, then I will perform the docking. When you will not able to know the structure, in which pocket you will perform the docking, then you will go with the blind docking, and blind docking is not easy to publish. So targeted docking is required. For the targeted docking, you have to read the literature carefully, whether your protein is receptor, whether is your protein enzyme. So accordingly, you have to perform, you have to design your docking study. For example, I have downloaded this structure, and it can be opened via the Chimera software. So just open this structure with the Chimera. It will take some time. approximately 30 seconds, 30 seconds. Now it is open. So this is the structure of NOS. So where we should perform the docking, where, how you will get the clue. For example, you can modify this molecule in hydrophobic surface area, and here you can see the various type of grooves and pockets present in this protein. But when you rotate this protein structure, where is my area of interest where we should perform the docking? So that thing, you, when you will do literature survey, then you should know that there is the heme domain that is called catalytic domain also. So this heme domain will be my the interest, and there is, if you go with the chimera, then chain, there is 2 chain, and it will be appeared here. So here 2 chain, A and B, and both chain, the sequence of both chain is similar or identical. So we have to remove one chain, any one chain you can remove. So for example, I have deleted chain, selected chain B, and we will delete the chain B from here. Okay, so we have deleted the chain B. Now you go with the residue. So here there are 4 or 5 type of residue: H4B, heme domain, HOH, OUM. So what is OUM? So you will get the clue of OUM from the literature survey. OUM is this molecule. This is the inhibitor molecule. So first of all, when you have decided that our target is enzyme or any protein, then you have to search your structure with keyword inhibitor, and that will give you a clear cut instruction where you should perform your docking study. So here, when we select the OUM, you should know that this is the small molecule, small molecule is bonded in the heme domain. This is the heme domain, this is the heme one, and the small molecule or inhibitor is bonded in the heme domain. So we have to delete this molecule first, and otherwise your molecule will interfere with this molecule and cannot be docked here. So you have to remove the heteroatoms. So you have removed the crystallized water content. If crystal water is present, then you also have to remove the crystal water molecule, because the crystalline water can also impose some error in the docking. So we have what type of molecule we have to remove. For example, you have selected the heme, and you should not remove the heme domain, because the heme domain is necessary for the docking. And when you will remove this heme domain, then you cannot, your docking will not be accurate. So you should, what molecule should be kept in the structure, and what heteroatom should be removed from the structure is called protein optimization. And that all thing, that the clue of that information you will get from the literature of that is deposited with or associated with this PDB ID protein. So you have to learn very carefully during designing of your experiment. So now I have deleted. Now this molecule can be saved. We can save this molecule from here. For example, I have saved molecule as a PDB file. You go with the file, save PDB. So you just save PDB. So before saving, you have to clear selection, means all molecule I want to save. So just save PDB, and where it will be saved. You can define the path also where you want to save this molecule. So for example, this is NOS. So we have put nos.pdb and it will be saved on the desktop. OneDrive desktop. So use and transform coordinate and just save. Here you can also by using Chimera, you can also able to modify by you just press the control and just select the residue. You can delete this residue also. So Chimera can be learned. When you will operate with the Chimera, you can learn the Chimera. So this session has 2 hours, so we cannot learn all the thing, but I'm just performing the docking. I have saved the PDB structure. You can also go with the tool and you can perform the structure editing, and here you can build the structure, you can minimize the structure. So the structure minimization process can be carried out before the docking. That will give you accurate result. So structure minimization will take about it will take time depending on your configuration of your computer. So when I will click minimize, it will take about 30 minutes to minimize the structure. So I will not go with this step. Now we have downloaded the structure of a small molecule. We have also downloaded the structure of our protein. Now we will perform the molecular docking. How we will perform the molecular docking? So you just go with the Autodock software, just open the Autodock software. You can also perform the virtual screening using Pyrex. So there is a limitation, but I am showing you. Okay, so when you open the Autodock, it is open now. So after installing the MGL tool and Autodock 4.2, you will see this type of window. Now we have to take ligand from here. So our ligand is in the SDF file you have downloaded. If you have downloaded from the file from the PubChem, then it is in SDF format. So structure format can be converted using Marvin sketch to the PDB file. So you have to convert first in the from the SDF to PDB, and after that you have to convert that PDB into the PDB-QT file, because the Autodock will read only .pdb-qt file. So in the input, you just open your PDB file. For example, I am going with the aldose. So here, for example, PDB is not there. For example, this one, this is the not PDB. It is also not PDB. Not PDB. Okay, this one. This is the PDB file. So when you will go with the for example azithromycin and PDB file, you just select the PDB file of azithromycin. Now it will give you some it will automatically add some gastrial charge. It will merge 67 non-polar hydrogen. It has detected 12 rotatable bond, and it is torsive value, means it has a 12 rotatable bond, so its torsive value is 12. Now okay, so now you have to output save as PDB-QT. So just go with your where you have saved your molecule in the folder name Autodock. So you just go with the Autodock, and here we will save our molecule, for example PDB-QT. Now I have made converted our PDB-QT here. Now this is the ligand file. Now you will go for the grid. Then in the grid section, you just click on macromolecule, just open it. Macromolecule file is all type of file, and here PDB is there. So you have downloaded your protein file, or you have saved your refined structure in .pdb file. So you have to upload .pdb file in this window. Now it is uploaded, and it will be automatically, it will prompt to the Autodoc. So you just write the name of protein .pdbqt and just save in the your respective folder. So now we have two more molecule in our folder. One is ligand molecule, like our ligand is azithromycin, and okay, grid macromolecule open.
Yeah.
Okay, yeah, okay, this is here. This one. So this is our macromolecular file, and this is the small molecule file. So this is the protein, and this one is small molecule. Now we are ready to perform the grid generation. For the grid generation, for example, if you know that you have to perform your molecular docking study, where your molecule should be bonded. Okay, grid macromolecule, choose select molecule. We have again saved. It is no secondary structure. Okay, now we can delete all the molecule. If it is showing some error, then you delete all molecule. You can again upload the molecule. For example, this is our PDBQT file. No. Okay. Now here Autodoc give you some option for the visualization also. So you can convert this molecule into the ribbon structure, like visualization in Chimera. So for example, we know that our small molecule is bonded here, for example. Then we have to set map type, and you will choose or open your ligand. So our ligand is azithromycin, for example. Then we will load the azithromycin here. Then we will go for the grid box formation. Now grid box formation is the critical step. So your grid box should be focused, should be placed at that place where you want to perform your docking. So when you will enhance the grid box size, that should encompass the catalytic residue or active site of the protein where the docking should be performed, where the small molecule should be rotate or bound to the protein pocket. So you can also move this in the XYZ direction according to your need, according to encompass your all active residue. So after that, when you will increase the size, it will take more and more time for the computation, for the docking. So if catalytic site is too large, the molecular docking time will be increased. So now we will save this molecule, close saving current. Okay, so we have saved this molecule, we have saved the grid file, grid box, we have generated the grid box, and now we have saved our grid box. Now we will set our output file as a GPF. GPF means grid parameter file. So now we are going to save our molecule, and I will recommend you to save your file with the name of your ligand. Our ligand is azithromycin, so I am just typing azithromycin. Azithro. So azithro.gpf you have to put. Azithro.gpf means grid parameter file you have saved. Now you can check in this folder also. You have generated another file gpf. You can also in the next step we have to generate a file dpf, means docking parameter file. So grid is finalized. Now we will move for the docking step. So now docking for the molecular micromolecule, we can perform two type of docking: rigid docking and flexible docking. So in the rigid docking, your micromolecule structure will be rigid, and your small molecule will move with various conformation. So I am just clicking on rigid file name, and you have to select your macromolecular file here. Then you have to choose your ligand. Your ligand is erythromycin. You have to select, just accept the default parameter. Then you just go for the search parameter like genetic algorithm. Here we have to understand, because in the search parameter there are three type of algorithm is available for the in the Autodoc. That is genetic algorithm, simulated annealing method, and local search parameter. So I will prefer the genetic algorithm method based on the literature. So when you click on the genetic algorithm method, here is the some parameter you can change, like number of GRN, how genetic algorithm, how many run you will perform. So this is directly linked with the generation of conformation. Means, for example, if I will put this value 1, means your molecule will dock in only one conformation. When you will increase this number like 5, then your molecule will your small molecule will docked in the grid specified area with 5 conformation, different 5 conformation. So Autodoc developer recommending at least 50 runs, but if you increase the runs, there will be more computational time. So here for the limitation of the time, I am preferring 5 GRN. So I am giving the 5 GRN and accept. Now the docking parameter. The docking parameter, you can also accept the default docking parameter. After that, you should go with the output. So which type of genetic algorithm you will prefer? There are 6 type of algorithm is there, like Lamarckian genetic algorithm, like simple genetic algorithm, simulated annealing, local search, evaluated energy, Vina configuration. So I am going to Lamarckian genetic algorithm. So I am saving our file in the genetic algorithm for the genetic algorithm, Lamarckian genetic algorithm. So now we will put azithro.dpf. Here dpf means docking parameter file. So we have saved docking parameter file. Now we are ready to dock. If you are using Linux or Unix, then you can run from here, run AutoGrid, run Autodoc, run Autodoc Vina. But for the if you are using Windows, then you have to download the Sysbin and you have to open the Sysbin. So you in the Sysbin parameter, you have to locate your folder. You have to give the command to the Sysbin to locate your folder. For example, change directory cd is means change directory C. We will go first in the C drive. Then cd, we have moved to C drive. Now the change directory users, okay, / if your computer has admin, then you put admin, and I have renamed my computer in SBG SDS. So I will put this one and Autodoc. In the Autodoc folder, our all compound is located. So here the command has received to the Autodoc. Now we will give an two command. To first we will run AutoGrid from here. This one AutoGrid. We should first we will run the AutoGrid. After that we will run Autodoc. So by the Syswin, using Syswin, first we will run the AutoGrid. For the running of AutoGrid, we have to give the command like dot, note carefully, dot slash AutoGrid 4.exe. This one AutoGrid 4.exe minus p, and the name of your GPF file, because we are running grid file. So we will name the GPF file. So our GPF file name is agthrow.gpf minus L and agthrow.glg, and this is the command for the AutoGrid running. So when you put this one, you can see, you can able to see there are too many files automatically is running. When AutoGrid will be completed, it will show done after pressing the enter button. And you can also see here in the meanwhile, I am, due to the time limitation, I am giving you the command of Autodoc. So after completion, I am assuming that our AutoGrid has been finalized. And what will be the command for the Autodoc? The same command will be utilized for the Autodoc. So for the Autodoc, there will be some slightly change, like here instead of GLG, it will be DLG, and here it will be, instead of GPF, it will be DPF, and here in place of the AutoGrid, it will be Autodoc. Autodoc 4.exe agthrow.tpf minus L agthrow.dlg. So DLG, what is GLG? Grid log file. We are converting grid parameter file to grid log file. Here we are converting our docking parameter file to docking log file. When you will convert, you will made the DLG file, then your docking has been finished. Now you have to analyze the docking result. So for example, we are assuming that our Autodoc is also run and form. I am giving an example. For example, this one, there is DLG has been made, and here GLG is also made. So you can open your DLG file, open with Notepad, and you can get several results. For example, this one. Here one table will be given in which the best conformation, and you will find here lowest binding energy. -9 is the lowest binding energy of azithromycin in our protein target. So minus 9.8 kilocalorie per mole, and it has the Ki value of 65.9 nanomole. You can copy and paste your result for the research purpose. Now you have to compare the binding energy will be lowest, then it indicate directly proportional to the binding affinity. For example, your small molecule, reported a small molecule which is available in market or reported as inhibitor, has you have to perform docking with that molecule also. And for example, that molecule, for example, beta-glyptin has is our control or reference molecule that has binding energy -7, but our molecule of has -9 kilocalorie per mole binding energy. Then I can defend ourselves and I can present our molecule as a lead molecule, and we can say that our molecule means azithromycin has higher binding affinity toward the target as compared to their reported inhibitor. It is clear? Is it clear?
Yes, sir.
Okay. Now, on the basis of DLG, we can open the DLG molecule in this window. So you just go to the delete all molecule and just analyze your molecule, analyze your docking result. You just open here azithromycin DLG. You should load the molecule. Now we will load the micromolecule. So this is our micromolecule. So now you can check when the first step, second step, after that conformation, play ranked by energy. You just click play ranked by energy and just convert this file into a ribbon structure for the better visualization. So this is your molecule. Okay. Okay, this is your molecule. Are you able to see this molecule?
Yes, sir.
In the wireframe. So azithromycin is embedded here where we have performed the grid. We have located the grid. So here azithromycin, you can play the conformation of azithromycin and you can see. So one conformation, second, third, fourth, five, six. How many the conformation is dependent on the genetic algorithm run parameter. This parameter, the docking parameter and the search genetic algorithm run parameter. So if you have put 50 GA run, then it here 50 conformation will be available. Now we can check here the lowest binding energy. I just open the panel to show info and to build the hydrogen bond. This will give you an insight into the hydrogen bond formation. For example, our molecule azithromycin is bonded to the target molecule and formed one hydrogen bond with tyrosine 310. So whether this molecule is critical or not, you have to decide. So your molecule is interacting with the critical residue or not. So for example, this is the least lowest conformation, and we want to write this lowest conformation in PDB file. Then you have to build current, click on the build current and write complex, and your complex will be written complex 1 dot PDB. So now you can save this one. So we have saved our conformation complex 1 in the Autodoc file. Complex 1. Okay, where I have saved this one. No? Okay, complex 1. So you can also visualize this complex in the with the help of Chimera. So you just open the Chimera and you can visualize and you can establish the interaction with like this. You can preset like interactive ribbon. You can preset like a publication type of data. So this is the this one is your azithromycin and it is embedded into the active domain or catalytic domain of the protein. Now we have to draw a Lig plot. You can generate 2D spectra of that molecule by a Lig plot. So just I have installed the Lig plot and not able to find Lig plot. Lig plus, yes, Lig plus. Lig. Okay. Okay, no problem. Yeah, yes, sir, yes, sir. Okay, where I have. Okay, this one, sorry. This is our link plot. I have installed it. Now you just retrieve, open, go to the file, open PDB file and browse. And we have put our molecule in the C drive. C drive, users, this Autodoc. Now this folder is erythromycin and PDB file. This one, complex one, is open. So this will open and you just click on run. Now the structure is ready for the publication view, and after running you can you can see that you can edit also here. There are several type of option and on off. You can increase, decrease the size. For example, if you want to emphasize on ligand residue name, then you have to put here for example one, then it will show like this one, the size has been changed. So you have to arrange all the file. You can move the residue also. So there is 2 hydrogen bond is formed with our molecule azithromycin with tyrosine. This is the tyrosine 310 residue that appear in the window of Autodoc, and this is your azithromycin residue. You can right click here and type azithromycin. For example, this one you can and you just rearrange the hydrophobic interaction. You can arrange like this. Okay, so this is all about the molecular docking in very brief. By practicing it, you can perform. Of course, there are several tutorials also available in YouTube. So any query? Sorry, I have taken more time, sir. Sorry for that.
कोई बात नहीं सर, एक्चुअली इतना तो टाइम लगता ही है अब जब हम जाते हैं डॉकिंग पे सॉफ्टवेयर में काम करने के लिए।
यस सर, यस।
और ये एक दिन की बात नहीं है, सही बोल रहे हैं आप, प्रैक्टिस की बहुत जरूरत है।
यस सर, यस।
है ना, तो ये ब्रीफ इंट्रोडक्शन विल हेल्प द पार्टिसिपेंट,
and it certainly motivates some participant to do the molecular docking analysis, and he can publish in UG. He can also publish a paper in Scopus Index Journal.
Okay, sir. May I conclude today's session?
Yes.
With permission.
Yes.
Thank you, sir.
Okay, thank you so much, sir.
My heartful thanks to our speaker, Dr. Gaurav Kumar, sir, for sparing his valuable knowledge and the time. Sir, you are very well explained how we introduce the drug in the computational method in drug design. You also explained pharmacophore, the relevance and method involved in the improvement of binding interaction between drug and its target. And you also explained finding the lead compound, role of combinatorial chemistry, drug action at receptor, target action, improvement of pharmacokinetic property of drug. So this is all about the computer-aided drug design, which can help the participant to make a new drug. Basically, these are based on the AI as well. So you have about the knowledge about something in computer The sir will explain what is the different window, that is a different operating system, Windows OS and then Linux. Sir, you are really genius. You have a number of papers for that number of this work you have done. So I would like thank you, sir, for
Thank
sparing
you
your
so much,
time.
sir, for providing this great opportunity. And dear participant, you can note my number also. You can be touch if you have any required any assistance or any guidance, then you can also reach to me. So sir will share my number also.
Okay, sir.
Thank you so much.
I extend my thanks to our respected chairman, sir, Engineer Sanjeev Agrawal, Vice Chairman Prashant Jain, sir. I would like to thank our director, sir, Dr. R.B. Goswami, sir, for his constant support, and he gave us a lot of encouragement. Thank you, sir. Last but not least, I appreciate every participant who joined and spared their valuable time. Basically, हमने 2 घंटे का टाइम दिया था और 3 घंटे ऑलरेडी हम ले चुके हैं पार्टिसिपेंट के. Thank you for all. I hope you see you all tomorrow for second day program. Tomorrow we have Mr. Vinod Devraj ji, trainer from the Sodinger, to explain the Sodinger software as well. Thank you again, sir. Thank you all participants. So with due permission, I am going to end this session.
Yes, sir. Yes.
Thank you all.
Thank you, sir.
Thank you, sir.